one of the systems my lab still works with, which is looking at fruit flies as this model for accelerated aging and thinking about the metabolic effects of that type of selection. But I'll sort of give some. Okay, kind of give some background to orient everyone who might not be familiar with experimental evolution or using experimental evolution to test the evolutionary theory of aging. So briefly, experimental evolution is kind of a relatively simple idea. You have populations in the lab, right? You bring them in the lab, then so you can imagine you have your control populations, things are all nice and good. And then you decide, okay, I'm gonna derive some populations from them, stress them out some way, right? So in this cartoon, we'll say we're introducing some sort of maybe a toxic element to their diet, right? And then we basically study how these populations respond to these conditions over many generations. So you can kind of answer questions about, what are the physiological adaptations that are allowing them to, say, process this toxic food better? And then you can also just think about general evolutionary questions, about the rate of adaptation, repeatability of adaptation, and so on, right? So sort of classic population genetics, evolutionary genetics questions. Over the past, I guess, 15 years now, people have really been incorporating sort of molecular techniques into this work. So we sometimes call these evolve and resequence experiments, where you're going to do a lot of genomics of these populations as they're evolving. So then you can study the genetic basis of adaptation, both in terms of the underlying dynamics and which alleles are responding, right? And you can also do this with transcriptomics as well. So that's kind of the general area where a lot of my work has been with fruit flies and with yeast, right? And then just to sort of orient everyone to the types of dynamics I'm looking at and I'm interested in, when you think about experimental evolution, you can kind of split it into two groups. You have asexual systems. These are things like E. coli and yeast, where you're primarily studying adaptation from new mutations, right? So kind of getting some of your classic selective sweeps and all that fun stuff. But I don't really do any of that because I don't like that. So I mostly work in sexual systems where adaptation is going to be primarily from standing genetic variation, right? So those are the dynamics we're interested in. So every time we see fruit flies, I'm not working with inbred lines, right? We're having these large, relatively outbred populations. Otherwise, we wouldn't have the capacity to see any adaptation, right? So kind of your big picture overview of my experimental approach. In terms of the system I'm going to be talking about today, this is based on work that my PhD advisor, Michael Rose, did. And we've sort of, various former students have inherited parts of these systems. So I figured I'll talk about the questions the system was created to address and talk a little bit about the evolutionary theory of aging, just in case anyone's not super familiar with that, right? So the question is, we all know, you experience physiological decline as you get older. And this gets worse over time. So like 15 to 25, not that bad. 25 to 35, kind of bad. And then it keeps getting worse and worse as you get older, right? So it's sort of like, why does that happen? And this sort of figures, sort of a synthesis of work from a bunch of different people, George C. Williams, Medawar, et cetera, about like why we think this happens, right? So I'll just briefly go through this. So here on this plot, on the y-axis, we have, the force of natural selection acting on a given individual in a population, right? And then on the x-axis, we have different ages. So the idea is prior to the start of reproduction, natural selection is acting at full power. And then after that, there's going to be a decline in the force of natural selection acting on that, or those individuals, right? So to kind of give a simple example of why this logic kind of works, you can imagine if you had a dominant, lethal mutation that had the effects were felt during infancy. That allele has no ability to get into the next generation because individuals are going to die before it can be passed on, right? So its prevalence will basically just be the rate at which that mutation occurs. However, if you have a dominant lethal, but let's say we're thinking about humans, and the effects are felt when the individual is like 60. By 60, you've likely already reproduced, you've passed that allele on, so it's going to be persisting in the population at a higher rate than that, right? So when we're thinking about the evolutionary theory of aging, one big aspect of it is this idea of mutation accumulation. So because of this difference in the strength of natural selection, at like the age-specific difference in the strength of natural selection, over time in populations, you should have the buildup of these deleterious alleles with late-acting effects, right? Because you can't effectively get rid of them. So from that perspective, aging is basically, as you're getting older, more and more of these alleles are manifesting in terms of their effects and you're deteriorating, right? Sort of related to this idea, and also thinking about how you can get a lot of these alleles is thinking about antagonistic pleiotropy. So as a result of this dynamic, now if an allele has a beneficial effect early in life, even if it has a potentially big cost or any cost later in life, selection will still favor that allele because you're getting its benefits when selection is the strongest and most important. Obviously, these two aren't mutually exclusive, but the idea is because of these types of dynamics, we have all of these late-acting deleterious alleles in populations. So in terms of doing experimental tests of this, you can just sort of think about, you have this big parameter, right? The start of reproduction. So you can say, if I modify or artificially shift the start of reproduction, I should be able to create different patterns of aging and longevity, right? So if you wanted to create populations that live longer, it should be as simple as delaying the start of reproduction, because you're essentially extending the amount of time where natural selection is acting at full power, going to weed out more of those deleterious alleles, right? And this is essentially what my PhD advisor, Michael, and then there's another guy, Bill Luckenbill, sort of did these experiments at similar times, where they were like, hey, this is actually pretty easy to do in fruit flies. So oftentimes in labs and fruit flies, we maintain them on non-overlapping generations, meaning only eggs laid on a given day or a given time period are used to found the next generation, right? So here we have this cartoon showing, we have some baseline types of populations where only eggs laid on day 14 are used to go to the next generation. So as far as natural selection is concerned, that's like the only day, right? That's the start, and right. So everything else should revolve around that a little bit. So all you have to do is like collect your eggs a little bit later, and then you should be able to test the theory, right? So kind of a simple idea. So this is effectively what they did. They kept delaying or pushing for many generations the start of reproduction all the way out until they weren't doing egg collections until day 70, right? So they kind of did this for quite some time. And then this is kind of some of the original survivorship plots from this experiment. So you can have in blue line, this would be the mortality or the survivorship curve for the normal population, so percent surviving over time. And then you have the old selected ones, so they're living longer, and the actual mortality pattern is different. You don't get the same immediate acceleration or quick acceleration. And then you can also go in the opposite direction, right? You should also have the prediction that if you shift the start of reproduction, to the left, you're now reducing the amount of time where natural selection is acting at full force. As a result, you should have populations where you're having more of this mutation accumulation. You might have more intense antagonistic pleiotropy sort of situations occurring. So this was also done. And then we went all the way back to day 9. You did start running into like some hard limits where you just can't get them to pupate sooner than this. Because I think the dream was like a 7-day cycle, because it'd be really fun for maintenance purposes. But like 9 to 8 seems to be the limit. So there's still some underlying contingencies and limits here. But this is actually a pretty extreme difference. Most flies in the lab are not even adults. They're still in their pupa during this time period. And then here, when we look at the survivorship curves, you see you get the expected patterns where the populations, they have much shorter lifespans, dying a lot faster, et cetera. So this is the primary system I'm going to be talking to you guys about today. So here we have 2 sets of populations, 10 replicates each. So we have our A-type population. So this is where we've had this intense selection for early reproduction. And then their controls are kind of on a much more relaxed selection where eggs are only collected on day 28 for the next generation. I'll talk a little bit about this in a bit, but we do have like some subgrouping within the system. So this is why you see different generation ranges. So within the A and C classes, we have new and old versions. We created new versions to test some hypotheses about evolutionary repeatability that I'll talk about in a little bit. So that's just why that comes up. And then, yeah, you can see some details, right? So we have them in this. If you go to my lab, it's just full of these plexiglass cages full of flies. So we can kind of keep them outbred and have that variation. Okay, so next I want to talk about, aging. So we're always going to talk about trade-offs. So it seems like a lot of people in this department don't find trade-offs with aging. So I don't know what you guys are doing, but I find them. So we're going to talk a little bit about some of the phenotypes associated with these A-type populations, right? So as we talked about, you have reduced lifespan, and then you have accelerated mortality rates. So this is looking at log mortality for females, males for the A and C types. So some things that jump out immediately, right? So literally by the time you get to around day 50, all of the A types are dead, whereas the C types keep going on until day 70, right? So much longer lived. And then the actual mortality curves themselves are quite different. If you're thinking about, sometimes you can think about you have the aging phase and the non-aging phase, if we're defining aging by sort of an acceleration in mortality. So the A types essentially start aging right away. In the C types, you sort of have that delay before they actually start aging. So this selection has created some really distinct patterns by that metric. Then we get a whole bunch of other stuff correlated with this. So overall, obviously, over the total lifetime, the C types have way higher fecundity, right? So this is looking at female fecundity. So, and I will say, I always do age from egg. I know sometimes if you've seen a lot of Drosophila talks, people do age from when they eclose. I just like egg better. So yeah, the C types have greater total reproductive output, but there is an interesting pattern, whereas essentially the A's have like really gone all in on early reproduction. So you have these first few days of adulthood where their reproductive output is just higher than anything else we see in the lab, but then it kind of crashes after that, right? So we would say this is sort of, you're seeing that optimization around that early reproductive window, and then you're seeing an overall trade-off where their lifetime reproductive output is much, much lower. We also see that this changes developmental patterns quite a bit. So selection, for early reproduction, so you're able to reproduce early, you develop a lot faster. So this is looking at pupation rates. So this is age from when they hatch from the eggs. And you can see you kind of have, by around, getting to our 80, 90, whatever, the vast majority of the A types, if we're thinking about the females, have sort of pupated, whereas it's sort of shifted in the C types. And this is one of those things if you were to ever work with these in the lab, you would just notice that the C types literally just don't exist as adults for there's a 2 to 3 day period where most of the A's exist as adults and they don't. So in my mind, that's actually a pretty extreme difference. And then we also have just general reduction in stress resistances between them, right? So some of the classic ones, this is looking at adult flies, so around, or mature flies. So these are around 20 days old in both treatments. We have reduced starvation resistance, desiccation resistance. We've also done some different measures of physiological performance. For instance, with cardiac arrest rates, you literally, strap a fly to a slide and you zap them, and then you see if their heart can regulate. And I don't know who came up with that. It works, but for whatever reason, the C types go into cardiac arrest way less than the A types, so their hearts are more robust. So they have this potentially increased cardiovascular function as a result of this. So when we think about the A types, I sort of make the case that this is a model for accelerated aging, right? Where we're having the early reproduction, they develop quickly, and they have this early fecundity boost, but then they basically fall apart, right? So they die younger, decrease lifetime fecundity, stress tolerances, et cetera. So, when we compare two cages, we would basically say, and this is all selection on standard genetic variation, it's almost like we've just got all the people who age really badly in a cage in the A types, and then the C types has the full range of different ways things can age. So kind of that enrichment for this accelerated aging phenotype is what I've been interested in. And the idea is by comparing and contrasting between these, we can learn about the aging process and what's underlying, in human populations, people aren't necessarily all aging in the same way. Some people have the rate at which their disease susceptibility and stuff like that is happening is occurring faster for some than others, right? So understanding that variation in aging in populations is part of the goal. Okay, so next most of this talk is gonna be on some of the recent efforts we've done with metabolomics in this system. But before that, I wanted to talk a little bit about why I decided to mess around with metabolomics. Because for most of my career, up until about maybe 30 years ago, I was kind of a genomics guy, occasionally some transcriptomics, and doing characterization of this, whether it was this, yeast, or whatever, right? That was kind of the area I worked in. And I found this was a really powerful approach for studying sort of questions I was interested in about the dynamics associated with selection from standing genetic variation. So these plots here, it's kind of an aggregate of a bunch of different studies I was involved in, where we're looking at genetic variation in the A's and C's. So here, this is all the SNP variation. So every SNP would be, a point of differentiation between the groups. We have transposable elements, deletions, duplications, and then this represents patterns of differential gene expression on 14-day individuals and day 21 individuals. So I think just to touch on some of the cool stuff we found in this, obviously we found that adaptation is pretty polygenic, which, at the time was cool. Now I think everyone acknowledges that, so less cool. But one of the interesting things, we always had this idea there was a really early paper in this system that Molly Burke published where she sequenced these A-type populations after at that time they'd had about 600 generations of selection. And there was still quite a bit of genetic variation in them. Like there was more than we would expect given like population genetic models. So it kind of emerged this idea that you're having, highly polygenic response to adaptation, but then you're also having the maintenance of genetic variation through some mechanism, right? So to test that experimentally, one of the things we did was we said, okay, we have these long-standing populations that have been under A and C type selection for a really long time. So if we go back to the ancestors used to create those, which again is another lab population, and rederive them, right? If you have all of these maintained variants in that original pool, and then you're doing it, even though we're doing it like literally 20 plus years later, adaptation should happen in the same way again. And we actually found that, right? We had this repeatability through time. So really strengthening the idea that there is some mechanism maintaining genetic variation in these populations. Or adaptation is less about sweeps and stuff like that, and more about just like shifts up and down in equilibrium frequencies for genetic variants, which was kind of cool. But then that's all I could really say, right? That it's complicated, it's polygenic. So then people would always ask me, what do you actually know about aging from this system? Like, what are the aging genes? And I'd be like, I don't know. And I had a summer where I was at the Cold Spring Harbor yeast course, where I was just like shamed daily for my lack of biological insight into the system. So I just wanted to think about ways I could do that, right? So then I decided to, this is coming with the rise of these like multi-omic approaches, right? So I've primarily worked at this level, but thinking about, okay, how can we incorporate proteomic data, metabolomic data, lipidomic data into these experimental frameworks to hopefully learn a little bit more about like what the molecular factors that are driving these differences in the phenotypes we're observing, right? Okay, and I will say I've been working on this for about 4 or 5 years now. So our first effort, and we'll talk about that in a little bit later, was published in like 2022. So what I'm talking about right now is based on work from the last 2 years that a postdoc in my lab, Dave Hubert, has been leading. So here we decided we're going to basically sample a bunch of different ages in these A and C type populations and then characterize the metabolome at those different ages. So have this trajectory of how the metabolome shifts over time in these populations. So you can see we sort of tried to calibrate our sampling ages based on a little bit about getting overlapping chronological age, but also calibrating a little bit to the mortality patterns in these populations. So if you look at these 3 time points, so this is day 21, 28, and I believe 30 something, I can't remember. Oh, it's on the next slide. These all overlap between the two. But then we also wanted like a late age for the C types, right? So we went all the way out to day 70, where at this point, all of the A types are dead, right? Then we also wanted a really early time point for the A types. So we did a day 9 sample. And at that point, we can't do sampling for the C types because they're still in their pupa, right? So we did, some details. We had to do a pooled approach, and so people approach metabolomics in a few different ways. You can try to do the smallest pool possible, and I think for people working in inbred lines, that's sometimes the way to go. We actually wanted to use kind of a larger pool size because we know there's quite a bit of genetic variation in these populations. So we wanted to try to sample that variation a little bit. So we settled on 50. Kind of calibrated to some other work we've done with other molecular stuff, right? Yeah, then we have total pool. So we did, again, in terms of metabolomics, you can approach this in 2 ways. You can do untargeted or targeted. So untargeted, you basically try to characterize everything your mass spec will pick up. So yeah, metabolites, just you have a tissue, whatever metabolites, so like sugars, proteins, whatever are present in it. There is a nice thing about untargeted where you're not biased in what you're looking at, but then your ability to do quantitative work is quite limited, right? It's really good for presence absence kind of analysis, but you can't really do good quantitative work. So we focused on a targeted approach. So this work was done in collaboration with some folks at University of Washington. So they kind of just have a panel. So targeted is you have a panel with standards, and by comparing to that, you can have better quantification of the relative abundance of the different metabolites. So their panel, I believe, is about 300 at the time we worked with them, and we found about 202 of those metabolites in our sample. So I think this is kind of an important point because this is not an unbiased reflection of the metabolome. It's a metabolome from a center that studies aging, that's grown panels for aging studies, right? So we have perhaps some enrichment for things that might be relevant to us. That's just worth thinking about. Okay, so first, we got all this data. We decided, well, first thing you do, you got a new omic, right? You make a PCA. So that's just what you do. So then we wanted to see how things are clustering here, right? There was some possibility nothing would cluster and the metabolome is not capturing the underlying physiology and then, be bad. But we actually find some interesting patterns of clustering. So if we look at the days that are overlapping between the two sets, so we have 25 to 35, we find that we do get distinct groups where we have these are all the C-type samples, and then these are all the A-type samples. When we get to these more extreme sampling points, one thing we notice is like the day 9 A-type samples are just really distinct from everything else, and we'll talk about that in a little bit, but they're kind of clustering all by themselves. And then interestingly enough, the day 70 samples actually cluster more with the A-type samples at this age, right? So this is kind of our initial suggestion that the metabolome is sort of capturing these true signals of the physiological state of the population and representing their age in some way, right? So, there's a difference. And then we sort of wanted to ask this deeper question of like, can we actually quantify or really test this apparent idea that the metabolome is capturing true signals of biological age? We did 2 versions of all of these analyses. There's a preprint up on this that you can see the full stuff if you're interested. But we did one version including day 9, and then we did one version of the analysis removing day 9. We ultimately decided to go with removing day 9 because this day 9, we were kind of hoping we gave them enough time that they had some time to eat, they'd had some time to do stuff. But day 9 is just very, very close to eclosion. And there's evidence in the literature that just because it's so close to eclosion, that might be driving why this day 9 is so different from everything else. And because we don't have an equivalent point to day 9 for the C-types in this particular study, it felt a little safer to drop that. I will say in terms of the qualitative results we get with or without it, they're basically the same general conclusions. We just felt leaving it out was the more defensible way to go about this. Okay, so the first thing we did, right, is the metabolome capturing signs of aging. I don't know how many of you follow the aging literature. A really big thing, people love clocks that like predict how old a thing is based on molecular markers. So we thought we could do kind of our own spin on that to try to tease apart if there is some capturing of these, or like if biological age a thing and is it being represented in some way. So to do this, we use an approach called elastic net regression. So there are probably other ways you could do this. This is just a common method in the field because, so elastic net, right? You're getting those aspects of ridge regression and lasso regression. So you can get some of the feature selection, but from the lasso aspect of it, but then you're looking at metabolites, right? So there's going to be a lot of collinearity between them. So having that ridge regression aspect of it is kind of nice for kind of finding the balance between those 2 things, right? So that's just a quick justification of that approach, acknowledging there are other ways we could have done this, and we didn't do a study on clock methods, right? So then our approach was to essentially make 2 big clocks, right? So we made a clock training on the A-type data and then a clock training on the C-type data. And then we have predictions about how the between regime predictions, right? So the idea is if the metabolome is capturing these signals of biological age, if you have the C-type clock, it should see the A-type samples as being older than they really are. Similarly, if you have your A-type clock and you apply that to the C-type data, if this is all working out the way we think it is, those populations should now look younger than they really are, right? So we sort of did that work. In terms of the within regime, we sort of did a hold one out method and then 5-fold cross-validation to sort of get to a good clock. And then you can use all the samples for the between, right? And then when we have our predicted ages and our actual ages, we fit this to a linear model for the different sets. So we can try to evaluate, like, are there differences in the slopes or rates of aging between them, right? So here is our results from the C-type clock. So in blue, we have the C-type predictions, and then in red, we have the A. So looking up here, we can see, if we train on C-types, it predicts C-types pretty well, so that's good. If that wasn't the case, this would be another point where we would just stop, but it worked out. And then we looked at C-types predicting A-types. And what we see is that at basically every age, the A-types are predicted to be older than the C-types. And the slope, right, of the line going through here is steeper, right, suggesting that not only are they being predicted to be older at every age, the rate at which they're aging is also predicted to be faster. So that's sort of our big conclusion there, right? Gets a little bit more complicated. We go the other way. Patterns aren't quite as clean, but we still get the same general in line with our expectation. So when we look at A-types, they predict themselves pretty well. You'd kind of expect that. And then generally speaking, the C-types are being predicted to be younger than the A-types, and the slope is a little bit shallower. So it's just not quite as extreme as the previous result. So from here, we would sort of, again, make the case that the A-type clock is seeing the C-types as younger than they really are, which again suggests, or at least our interpretation would be, this is suggesting that these data and these models are picking up on some true signals, right? So usually what people ask at this point is like, okay, what's in the clock? Are those going to be the secret to aging? Should I like eat a bunch of biopeptine or something? I don't know. But I would say don't do that, right? So what we do, so I'll talk a little bit about the interpretation of what's going on in the clock and then how we actually try to make sense of this, right? So, or we have this conclusion that the metabolome does appear to be capturing indicators of biological age. However, when we look at the metabolites being identified by these models, they're picking out different features, right? So we're using about 60 metabolites in the A-type clock and about 23 for the C-type clock. And then there was kind of the idea like, okay, you have different sets, but maybe there's some core set of overlapping metabolites that are your really good markers of age, and that should be showing up in both. We actually only find 4 overlapping metabolites, so no evidence of enrichment for like these like super predictor metabolites in the system. You could still, in principle, look at the metabolites being used in the clock and trying to interpret that, but we didn't really want to do that, mostly because ElasticNet still has this element of like dropping out variables based on if things are like highly correlated and stuff. So we don't actually think, like, there could be other things that are useful predictors in this model that would be part of these biochemical pathways or whatever, that we would still be dropping, right, because of our approach. So we didn't really think that was a sound way. And there's some literature in the aging clock literature about like not doing that because it can be a little bit misleading, right? Okay, so then we were left with, okay, what's our conclusion going to be? The metabolome is complicated, but it kind of works. So we didn't want to stop there because that's kind of lame and we'd just be back where we started. So we started up thinking of different ways where we could try to make sense of the metabolite data or draw inferences about the underlying metabolite or metabolic pathways. And this is kind of a challenge because one, we have a biased samples. If you're thinking about a given metabolic pathway, we don't have all of the metabolites within that pathway, right? So it's not as easy as just, going back to your biochemistry textbook and figuring out the inputs and outputs because you're missing some data. And then you also have the fact that a given metabolite can appear in multiple pathways. So it's not necessarily the case that you see something increasing, you can say, oh, okay, there's more of this particular type of metabolism occurring because you don't know the full structure, right? I'm going to show you 2 things we settled on that we decided to include in the paper, but some of the other things we tried was looking at, like, say, patterns of correlation between metabolites and so on to see if that could recreate networks, and then if it would tell us those networks are meaningful. Didn't work out. Oh, my battery's a little bit low. Hopefully that. Does anyone have a charger? Thank you. Good save. Okay, yeah, so our first approach is kind of, let's do the lazy approach. we'll do a linear mixed effect model and see if we can find sort of, metabolites that are showing really good, or where you have really strong effects of like selection, age, or the interaction. Okay. Yeah, so we sort of did that initially. This is kind of an upset plot showing those results. Unsurprisingly, most of our data set comes up as significant because we have a panel that was from a center doing aging work, so maybe not that surprising. What we can't pull out from this, though, is most of our sort of significant metabolites are dealing with either selection or age, and there's actually not a lot of interaction terms here. And then we have a bunch of them where you have selection and age, right? So you have a consistent effect that's different across age with them. So this is kind of nice in terms of reinforcing maybe what we learned from the clock, that there are these signals, right? Because you are seeing some consistency or repeatability to the underlying signal. So I think that's kind of what we took away from that. Trying to do enrichment and stuff based on these metabolites itself didn't really work out too well. Just some of the challenges associated with that. So then we were kind of in like another head scratcher mode where we had our meeting, because we outlined our goals, right? We want to find things about metabolism. I think myself underestimated some of the difficulties with that. But at that point, our sort of metabolomics guru at UW, he was like, you should make a giant heat map of all of the metabolites and then see if there are any cool patterns you can pull out. So first I thought he was joking. Then I questioned my decision and collaborators. And then I said, okay, I'm just going to do it. So we're going to see what happens, right? So we made the gigantic heat map. So this is it here. We can read it now. And then I'll pull out some parts of it, right? To sort of see these patterns. One thing we noticed was that, the way the sort of columns clusters actually did replicate what we think is happening based on some of these other analysis, where the C-type populations are like these intermediate ages, 21 through 35 cluster together. But then the day 70 for the C-type, again, clusters with the A, right? So this gave us a way to sort of narrow what we would look at. And we did this in a fairly naive way. So I'll say a lot of this is what we would characterize as hypothesis generating work for the future. And I will outline how we plan to test some of these hypotheses, right? So the first thing we could do is basically look for, so we're calling this, when everything is kind of clustered around here, we're saying this is like our aged phenotype where things are looking old biologically, right? So looking for things where abundances are higher in samples with the age phenotype, right? And then so we go through the clusters, we find two clusters where this is the genetic pattern. So we have cluster 6 and cluster 10. And then using this data or these metabolites, then we try to do some enrichment work, right? So this is the result of this enrichment work, right? And then what we see pulling out from the big terms, right? If we look at the age phenotype, it's associated, and again, some of this involves looking at the specific metabolites, are they up and down or whatever, is associated with increased activity in protein catabolism pathways based on a naive interpretation, right? So that's kind of our takeaway there. Then we looked at the other end, right? If you have your abundances are lower in samples with the age phenotype, and we get these three clusters here, so 2, 9, and 22. Then we do our enrichment games here. And now we find kind of another interesting pattern where there's a decrease in protein biosynthesis if you were to kind of look through these terms. And then the age phenotype is also associated with a reduced activity in carbohydrate metabolism pathways, right? So kind of the overall idea is that aging is associated with an apparent shift from carbohydrate to protein-based metabolism, and then this is happening faster in the A-type populations than the C-type populations, and this is driving some of this. So this is just a plot showing you what some of these actual relative abundance graphs look like. So this is looking at glucose, right? So, and I'll talk a little bit of how we interpret this, right? So if your glucose is low, then it goes up at another time point. The naive interpretation is you are using a lot of glucose here, right? And then you're using less here. So maybe there's more glycolysis happening here, less here, and so on, right? And then we can see some of the protein or the amino acids and stuff that popped out. And again, you see the pattern where it's higher or lower, right? The problem with this interpretation, why I say it's a naive interpretation, is we're just looking at snapshots. We're actually not, we don't know the actual tracing of like, did that glucose actually go into glycolysis? Or it could be as simple as like the C-type flies eat more than the A-types, right? So then they're going to have higher levels of these things. So to test that, or kind of next phase of at least this aspect of the project, are to do what are called flux assays. So this is when you're going to say, we're focusing a lot on sugars and proteins, right? So either your sugar source or your amino acid source, in the case if you're using glucose, you have with like tagged carbons in your glucose. So you can buy that, you can get small amounts of it. It's kind of expensive, but you would feed that to the flies. And then when you do your mass spec work, you can actually see where those sugars are going, or like those carbohydrates are going. So now instead of inferring that, okay, this glucose is driving, more glycolysis and going into TCA cycle and all of that, you would actually be able to trace this, right? And sort of see it in depth. This is a little tricky to do, so we're still figuring out the protocols and the appropriate way to do this, because you kind of have to have these weird loading phases. But that's kind of our next step. I will say we do have some general support from some work in this vein, where they were working with inbred lines and they compared very old and very young flies, and then they looked at tracing across different tissues. And they also found a pattern where, as when they compare old to young, the big difference was a shift from carbohydrate-based metabolism. In that particular line and environment, they saw a really big shift to like just breaking down like serine and valine or something like that. So a lot of protein catabolism. So that's kind of one area where we want to do some more work. And then we also want to think about, like, we don't want to purely just try to describe what's happening. Because we have, like, at this point, decades worth of data on these systems. How can we leverage that to actually generate hypotheses about like why we think this is happening, right? So we would say one of the big defining features when you're comparing A-type to C-type selection is this sort of absurdly high reproductive output the A-types have in that early window, right? And this is kind of looking at the glucose there again with the caveats. So and then, when we look into egg production in flies, the primary energy sources during that process is usually lipid and carbohydrate metabolism. We don't have lipid data, so we can't really address what's going on there at this point, but that's something we can do in the future. So the idea is, because of this really intense need to produce eggs, you're having this really high glycolysis or rates of glycolytic metabolism here. And then this is trickling down to different effects. So one predicted effect is like maybe a regular fly is like beefing up their energy reserves during these early ages. These would not be doing that, right? And here we do have some sort of corroborating evidence. This is from a paper where we were looking at physiological function in the flies. And as part of that, we looked at differences in energy stores in glycogen and lipids, so like just whole body measurements. And we do find that, and this is from day 21 individuals, that the glycogen and lipid stores are much lower in the A-types and the C-types at these advanced ages. So maybe giving a little bit more credibility to kind of this emerging story, right? And this is a result from my first attempt at metabolomics. So in this project, we don't have to look at it too closely, but we had sort of the C-types, the A-types, and then we also included another set of populations where we had selection for starvation resistance. And then we compared profiles at day 21. So we only had a single age. But one of the big things we noticed in terms of our differentiated metabolites is we had a lot of stuff involved with TCA cycle and then sort of the outputs from there. So as part of some of my other collaborations, I work a lot with a team at Vanderbilt and Penn State who do a lot of mouse and human work, looking at lipids and metabolites and whatever. And most of them are studying mitochondrial health. So when they first saw these results, they were like, oh, this seems like it's indicative of higher rates of mitochondrial dysfunction in your A-types and C-types. Obviously, they study mitochondria, so they'll have a tendency to think everything is mitochondria. But we decided to roll with it because it kind of makes sense, right? So one hypothesis we have is that because you have this really, really high rate of glycolytic metabolism at early ages, that is going to actually cause some stress to the mitochondria, right? Both in terms of oxidative stress, and then there's literature about, you have these intermediates being produced, like all this lactate and all that stuff, that's affecting pH in the mitochondria that can cause some level of mitochondrial sort of dysfunction or degradation. So the idea would be there's kind of this trade-off that's occurred because of this really intense selection for early reproduction, where you're sort of burning through all these sugars, doing all this stuff, and then you're almost like frying your mitochondria. And then this has these effects throughout your life that are also leading to some of this increased mortality and all that stuff. So again, sort of this idea that this high level of glycolysis is leading to that based purely on this result, right? So we're doing some work to actually characterize mitochondrial function and morphology in these populations. So we've done some early stuff looking at a few genes associated with sort of just mitochondrial function and morphology, right? And then this was sort of supplied a list from our collaborators of ones they thought would be good targets. So we see overexpression, this is based on qPCR, of ATF4 in the A types, and this is associated with mitochondrial stress. And then we see overexpression of mixed cysteine, morphology stuff, right? So we do have a little bit of evidence here. So right now what we're trying to do is sort of collect samples, because they do a lot of scanning electron microscopy. So we can actually look at the mitochondria and see how size is changing, differences in interconnectedness and so on. So to actually test a more mechanistic hypothesis about why these A types are aging faster or breaking down faster. Yeah, so I think that's about all I have. So I finished a little bit early. But the sort of our big conclusions at this point is that the metabolome does appear to be capturing true signals of biological age. And just within our field, I think in experimental evolution, people have sort of played around here and there with using some of these other omics, but there hasn't really been an in-depth usage. So that's kind of what we've been working on. And we think it's generating some value, right? We're especially going forward, we're thinking about, if you combine targeted and untargeted approaches, we might be able to get around with some of these bias issues. And this might be sort of a valuable way forward for any of our studies on complex trait variation to maybe get at some of the molecular mechanisms driving those differences. Okay, that's all I got. So these are my acknowledgements. Dave is the postdoc who's done a lot of the work, and then these are various collaborators and funding sources. So with that, I'll take any questions. We have done some transcriptomic work, but we haven't done in terms of the same resolution of the number of ages. So we've only done like 14 and 21. We do see that they cluster based on expression patterns. Yeah, that is one area that we want to go into a little bit more deeply of actually having these different data points at different time points. Yeah.
Do you have any indication on tissues that are contributing to those metabolic differences?
Yeah, no, that's a really good question. So we've been doing whole body to try to get these average effects. Some of our gene expression work, we've looked tissue specific. We do tend to see the strongest signals in the brain tissue and then the cardiovascular tissue. So I think that might be one avenue to really focus on that. I do think a tricky thing for me, because I think, and my students are all facing this because we're doing a lot of projects where we sample. When people do aging things and you have these measures, you want to make a clock. And then I'm just really don't know what it means if tissues have different clocks and stuff like that. So that's kind of one of my things I'm thinking about, if there's a way to use that to make sense of different patterns, right? Yeah. But yeah, no, good question. Something we haven't done but want to.
Does your metabolomic information suggest an experiment about supplements or something like that as a way of perturbing young people, or do you think it's been trained kind of thing that you can't push out of that's
Yeah. No, I think that's a good point. The unfortunate thing is some of it overlaps with things people already think would work as supplements. So one of the things, let's say we're proposing this idea of, the TCA cycle, mitochondrial dysregulation. So you'd have the idea that more antioxidants might help, right? And that would mitigate the effects in the C types, and it would have a disproportionately bigger effect on the Cs than the As. So that's one intervention we've talked about trying. There's also some these would get a little bit trickier about messing with egg laying versus non-egg laying if eggs are this dump for all of this extra stuff. So yeah, we're still thinking through manipulations that we can do.
Is it the thing to try to push the A's to C?
Or I guess push the C's to A's or?
Well, no, I was,
okay.
I wasn't sure what you
Yeah, so that would be the direction of if we try to give supplements to the A's to maybe mitigate some of the negative effects of that these trade-offs, they would have a more C-like lifespan. Yeah.
So following up on that, given the effects of exercise on mitochondrial health, is there a way to actually exercise lives?
Yeah. Yeah, actually, people do exercise flies, strangely enough. There's a paper on where they looked at high sugar diets, and they found that exercising flies mitigated that effect. So two ways you can do them is you put them on a string and you lift them, so when their feet aren't on the ground, they just flap. Or you put them in tubes and you keep spinning them, so they keep crawling up, right? So I'm not planning to do that. I will say one of my former lab mates, James, he just started his own lab in California, and he's more of a exercise physiology kind of person. So he's building these like weird spinny vial things. So he's hoping to like force the flies to exercise and see if that might mitigate some of these effects. Yeah.
So I hope you'll join us up in